DNA translation is the process by which the genetic code carried by messenger RNA, mRNA, is decoded to produce a specific protein. This process takes place in the ribosomes, located in the cytoplasm of prokaryotic cells, and either in the cytoplasm or on the rough endoplasmic reticulum in eukaryotic cells. Translation begins with initiation, where the ribosome binds to the mRNA at the start codon, AUG, signaling the recruitment of a transfer RNA, tRNA molecule carrying methionine. During elongation, the ribosome moves along the mRNA, reading each codon and matching it with the corresponding tRNA carrying the appropriate amino acid. As amino acids are delivered, peptide bonds form between them, resulting in a growing polypeptide chain. The process continues until the ribosome reaches a stop codon, UAA, UAG, or UGA, triggering termination. At this point, the completed polypeptide chain is released, folded into its functional three-dimensional shape, and directed to its specific role in the cell. DNA translation is a crucial step in gene expression, converting genetic information into proteins essential for cellular structure, function, and regulation.